Alright, so when it comes to microscopy and staining, there are two aspects to it, obviously. First part is microscopy, and the second part is staining. So when you start off with microscopy, the two definitions that are like pretty crucial to know are magnification and resolution. Magnification is the ability to enlarge smaller images, and resolution is the minimum distance between two objects where they can still be viewed as separate. So those are the two definitions that are pretty important to know for this section. Now let's look at some methods of microscopy. The three that we need to know are light, scanning electron, and transmission electron. So for light microscopy, let's just talk a little bit about the aspects of it and why we would use it and why we wouldn't use it, so advantages and disadvantages. So with light microscopy, it uses light. So just mentioned that it uses light. However, with light microscopy, one aspect is that the resolution and magnification are pretty low in comparison to the electron microscopes. Resolution, magnification, low. And this is because since it relies on light, the wavelength of light is quite long in comparison to electron microscopes. So it's not able to reach as much spaces, basically. The wavelength of light is longer than electrons. But one advantage when it does come to this is that you can investigate live cells because it doesn't have to be in a vacuum. So, and we'll come back to why the others have to be in a vacuum. So no vacuum, so can investigate live specimen. Another disadvantage is that there's no 3D imaging. And that's about it for light microscopy. So the resolution and magnifications are pretty low because of the fact the wavelength of light is longer than electrons, so lower resolution and magnification. It's also not in a vacuum, so you can investigate live specimen, and there's no 3D imaging, which is a disadvantage. And one last thing that we can actually say is that the specimen doesn't have to be thinly cut. So because it doesn't have to be thinly cut, again, you can use it to investigate live specimen. Investigate live specimen. It's often used to observe like actual processes like cell division, things like that. Let me just put red for disadvantage and then like blue for advantage. So this is a disadvantage. That's orange or red. I can find red, so closest thing. And blue for advantage. Also advantage. And then disadvantage. So that's the you know what, I'll actually even make a key here. So that's a disadvantage, that's an advantage. A little key to help us understand it better. Disadvantage, advantage. All right, okay, let's move on to the second kind, which is scanning electron microscopy. Scanning electron microscopy. That's really a mouthful, so we usually abbreviate it to SEM. Now the main advantage when it comes to this is that, as you can see in this image, it's a 3D image. So 3D imaging is the only one that can do that. So it's a big advantage because it helps us understand the dimensions better sometimes. And another advantage is that the resolution is higher and even the magnification is higher than light, but it's still lower than our third one. Resolution and magnification relatively high. Oh, and we didn't mention what it uses. It uses electrons. So because it uses electrons, again, it has to be in a vacuum. It needs to be in vacuum. So you can't really observe live organisms, so you can't observe live specimen. So that's your scanning electron microscope. And as for the advantage, this is the main advantage. It's like a big super duper advantage. This is also an advantage, but it's relatively, so it's like, I guess you could say it's green. And then this is a disadvantage. Alright, let's move on to our third and final type of microscopy that we would need to know. Now this is transmission electron microscope. Now this one also is a mouthful, so we usually abbreviate it to DEM. So this one has like ridiculously high resolution and magnification, so you can see very small things, so like your ribosomes and things. I was going to write ridiculously high. I'm not going to write that. I'll just write high. Let's write very high. Or actually, you know what? Let's just write ridiculously high. Let's be low. How do you spell ridiculously? I think that's how you spell it. I'm not sure. Ridiculously high magnification. Actually, no, that's not how you spell it. I'm not going to go back to that. And resolution. So you can see very, very small things. You can see like your ribosomes and like your lysosomes, like all the really small organelles. Much easier to see. I think that's how we spell it. But there's no 3D imaging, of course. It's obviously not an advantage. And then one other thing is that it's in a vacuum. And this one's like super high maintenance, so you even need it to be really thinly sliced. So the organisms usually die as a result because there's no, it's a vacuum and they're so thinly sliced. So the living specimen dies. Okay, in terms of the advantages and disadvantages for this one, this is obviously a big advantage, and then the other two are disadvantages. So they're all used depending on what the criteria is, like what you're really looking for. But that's like, those are like the quick overview of that. And actually, one other thing, like some people may wonder, like what's the difference between like how they both work in terms of the scanning electron microscope and the transmission electron microscope. I'm just gonna mention that really quick. So the I'm just gonna say SEM, which is the scanning electron microscope. SEM. It works based on electrons being scanned over the specimen. So SEM works based on electrons being scanned over a specimen. So they don't actually they're just like basically they're just over the surface of it. So for example, this was a surface of, this is a 3D surface, by the way. I hope that makes it look a little more 3D. And like this is your like point, like this, let's just say this is your microscope. That's a horrible microscope, but let's just say that is what it is. And it's like sending electrons, and it's just going to scan the surface of this and create an image, basically. Whereas with your transmission electron microscope, it actually goes through, which is exactly why the specimen has to be really thin, based on, it's in its name really for both of them, being transmitted through specimen. Alright, so that's that. Oh, and one other thing that I did mention that I didn't actually get back to was the vacuum bit. So why does it have to be in a vacuum? Why do electron microscopes need a vacuum? And the reason for this is because electrons, if they're, electrons are really reactive. Electrons are very high energy and reactive. So if it was in a vacuum, then they would like react with the other particles present, and it would not be effective anymore. Would react with other particles and be like reduced, I guess, with other particles, and make process ineffective. So that's the reason why it has to be in a vacuum. Very high maintenance, of course, but you know, it is what it is. We need good results. Yeah, that's about it for microscopy. Alright, so what is staining? It's the technique used to enhance contrast samples at the microscope level. So basically it helps us view the different things present in a microscope image, because often there are many things, and like it's quite hard to identify the different things based on just a black and white image, which is why staining helps with that. And there's like different kinds of staining. There's like well, there's two that you should know at like the basic level, I guess, and those are the two types. So one is histochemical. So let's talk a little more about this before we go to the second one. Histochemical is basically between, the staining occurs between tissue component and the stain solution. So to understand this a little better, there's two different types of this as well. So it says it in its name pretty much. Histo refers to tissue, and then like chemical is like the chemical that you're using to, change the color, which refers to the stain solution. Now there's two types of this. One is based on enzyme. This is pretty straightforward. So when an enzyme is present, it'll basically this kind of stain would detect that enzyme and then change the color accordingly. So an example of this could be alkaline phosphatase. And histochemical typically uses this thing called hemotoxylin and eosin. And hemotoxylin is a basic dye, but it dyes acidic structures basically, and it turns them deep blue or purple. Whereas eosin it's an acidic dye, and it dyes basic structures, so things like your cytoplasm. And as such, in this case, since we said alkaline phosphatase, obviously it works in alkaline conditions, which is why eosin would be used, and the color would become like pinkish. But we'll come to that. But that's how it works in principle. And that's what I'm going to get to right now, which is based on chemical. And over here we'll mention the big boys: hemotoxylin, I think it's like this, toxylin, and so this works. This is an basic dye, and it dyes acidic structures, such as nucleus, because the nucleus has deoxyribonucleic acid, and acid is acidic, obviously. So it dyes the nucleus, and it does the color it gives it is blue or deep purple, so it's like a really bluish color, but most of the time it's like really blue. And the other one, which is called eosin, and this one is an acidic dye, but this one dyes basic structures, so they both dye like their opposites, basically, basic structures. And so it dyes like your cytoplasm, for example, things like that. Alright, so that's histochemical. Let's move on to our second one, which is immunocytochemistry, but that one is I think that as well is in its name. I think the most important thing when it comes to these things is like looking at the name. I think usually the name usually gives it away, which is why medical terminology is such an important course, because it really helps you put everything in perspective when you restudy things. It's really helping me right now, because I would not have thought of this when I first started studying this, So as you can see in this, let's break it down. Immuno means immune cells. It means immune, right? And then cyto means cell, and then chemistry. So immune cell chemistry. So it's dying based on the presence of immune cells. Now, what are immune cells? Your antibodies, things like that. But in this specifically, it's usually antibodies only. So you could name this as, you could define this as using antibodies or, my goodness gracious me, wait. Or specific cell molecules, usually immune, because this is about immunocytochemistry, to detect different tissues. Those are the two types of staining. You can have a look at examples online, and that'll help you better understand. And that is microscopy and staining.